The KEYNOTE-042, first of all, recognized it was felt to be one of the top 4 abstracts of more than 6,000 submitted for this meeting. It was a very important study. It was looking at whether or not there are patients with non-small cell lung cancer who we would normally simply treat with chemotherapy, maybe add immunotherapy. And the question was, could we get by in some patients just using immunotherapy alone and not putting them through the rigors of regular cytotoxic chemotherapy? Now, it's important to note that everybody on this trial had some expression of PD-1. So there were no patients who had no PD-1 expression. But what we found is that in all groups, there was an improvement in how they did and an improvement in overall survival if they just received pembrolizumab versus if they got chemotherapy. Now, that impact was markedly higher the higher the expression of the PD-1. And there is some discussion whether some of their results are driven more by the number of patients with very high PD-1 expression, which made up more than half the patients on the trial. But I think it's something that potentially has huge impact in day-to-day care of lung cancer patients. So there were a couple of very important differences in those 2 studies and this study. First of all, those 2 studies only included patients with squamous cell carcinoma, and they also included all comers. They included a population of patients with no overexpression of PD-1, PD-1 staining literally of 0. And they found in those patients that putting the 2 drugs, the chemotherapy and the immunotherapy together, was better than just the chemotherapy alone. I don't think you can compare these studies head to head. So first of all, let me just say, to have this many good options for treatments for people with non-small cell lung cancer is something we could not have conceived of 5 or 10 years ago. Having, for lack of a better phrase, a wealth of riches like this means that we can even more precisely tailor the therapy to the person who's getting the therapy. If we have a person with higher expression of PD-1 who is a bit sicker, may not do as well on our typical chemotherapy drugs, we can feel very comfortable just giving them the immunotherapy alone and getting a good outcome from it. If they are a squamous cell non-small cell lung cancer patient and they have no PD-1 expression, we can still feel comfortable giving them typical chemotherapy with the immunotherapy, knowing that they'll do better. We are happy that our people under our care can have these kind of choices. As a matter of routine, now pretty much screen all of our patients for certain mutations, including EGFR, ALK, ROS. What's very important to remember, if you have a patient with an EGFR mutation, you should not think about using immunotherapy with them because it's simply not going to work. You need rather to use those agents that target the EGFR mutation. There was a study presented looking at plasma assessment of what's called circulating free DNA. That is DNA that's just floating around in the plasma, not associated with any particular cell. This is not circulating tumor cell detection. And what they did in their initial reviews is they presented a brand new methodology that they were able to screen out a lot of the false positives, and they found 51% of early stage lung cancers with an extremely high specificity. Now granted, our current screening for lung cancer, which is low-dose CT screening, has a higher sensitivity, but there's a lot of false positives, meaning of lower specificity. And unfortunately, we also learned at this meeting that for whatever reason, low-dose CT screening is just not being taken up as much as we'd really like it to. This was an early study on the circulating free DNA, but if the promise of this initial study holds, we could be looking at a day when you go into your regular doctor's office, get some blood drawn, and it's a screening test for lung cancer.